Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Before we dive into this episode, we have a few announcements to make. The program of the third edition of the Dr. GPCR Summit is taking shape. Our confirmed speakers include Drs. Andrew Tobin, Ryan Roth, Rosie DeWalibi, Karen Gregory, Oliver Hartley, Ines Liebscher, Christelle Manet, and many more. This year, the meeting will be held between October 10th and the 16th. The meeting will be virtual to allow you to join us from anywhere in the world. Live talks will be hosted on Zoom from October 10th to the 15th, and everyone is welcome to participate by presenting a poster or submitting a pre-recorded talk. Similarly to last year, we will have presentation prizes for trainees. We're also trying out a few new things as well. We'll have networking and poster sessions on Wonder. We'll also have a full day dedicated to trainees. If you're a trainee, hurry up and submit your abstract today to give a talk or potentially present a poster. Visit drgpcr.com/summit today. We will also be hosting 3 workshops. We're excited to share that we'll have Dr. Sam Hoare and Dr. Luciana Leo, who will run a workshop on data analysis. Dr. Nicola Smith will host a workshop on how to preserve data integrity in the lab. And this year, we welcome back the GPCR DB team with Dr. David Gloria, Dr. Albert Koistra, Gaspar Nandi Sekeres, and Jimmy Caroli. Everyone is welcome to attend the summit. And it's free when you become a Dr. GPCR Ecosystem site member, which is also free. Speaking of the ecosystem, we are excited to share that the Dr. GPCR Ecosystem 2.0 platform is now open. Visit drgpcr.com to explore the ecosystem today. Please note that we are slowly migrating from our old website to our new website. You'll always be able to find us at drgpcr.com, and we thank you for your patience during this transition period. The ecosystem, if you're wondering, is your GPCR-focused virtual playground. Join over 75 of your peers who have already registered as site members. Remember, it's free. You'll also have the option to select a plan and get access to all things Dr. GPCR and much more. You get access to the new podcast episodes with video before they get released to the general public, our new group discussions and forums, our job board, our learning center, where you'll be able to take a course or even prepare and share a course with your colleagues. And last but not least, you'll be able to discover GPCR companies and much more. Take advantage of everything that the new GPCR dedicated online playground has to offer today by visiting drgpcr.com and becoming a site member. Remember, it's free. Once you are a site member, you can also choose to sign up for an ambassador program, which means that you'll get your own dedicated Dr. GPCR ecosystem link. And every time someone subscribes using your link for a year, you'll get compensated. Last but not least, we're also looking for content creators. Subscribe to the ecosystem and start writing your own GPCR-focused content. Share it and show off your talents. You can always reach us at hello@drgpcr.com or visit drgpcr.com to find out more about all our activities. And now, let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR, and today I have the pleasure of having with me Dr. Patrick Sexton from Monash University. Hi, Patrick.
Hi, Yamina. How are you going?
I'm great. I'm so excited that we got to talk today. I have to ask, what time is it?
Oh, it's 9 a.m. It's a good time.
It's a good time. Okay, it's not too bad. It's 7 p.m. here, so it's not but it's already Wednesday, which I still find it fascinating. The time, working with the time difference.
Yeah, it's a joy when you've got meetings with people in the US and Europe and try and find a time. Most of that means I'm 2 a.m. in the morning.
Wow. And I remember last year at the summit, I think it was 4 a.m. when you were presenting. So kudos.
Quite possibly, yes.
Kudos to you. All right. So would you please give us a quick introduction as to who you are and what do you do?
So I guess I trained as a pharmacologist at the University of Melbourne, and then basically did my PhD at one of the teaching and research hospitals linked to the University of Melbourne. I I would classify myself as a molecular pharmacologist, and myself, or more correctly these days, my research group studies the structure and function of G protein coupled receptors, not surprisingly, with an emphasis on the class B1 peptide hormone subfamily. And within the team, and the team is co-led by Professor Denise Wooden, who I've worked with for many years now. We have projects studying GPCRs across most areas of preclinical research. So from elucidation of structure and dynamics, structure, function, patterns of signaling and regulation at cellular level, including, obvious things like biased agonism and allostery, through in limited projects to whole, animal physiology and models of disease in some cases. And I guess I work as a research-only scientist. So my position is as a senior principal research fellow funded by one of our major funding bodies, the National Health and Medical Research Council. I have an appointment as a professor within the drug discovery biology theme at our institute. And I'm also the director of a training centre funded by the other major funding body, the Australian Research Council, and that's in the area of cryo-electron microscopy and membrane proteins.
Wow. And how do you juggle all of these together? It's a lot of titles.
Well, I'm very lucky, in being able to have Denise as a co-leader of the group. Denise actually does a lot more work than I do in keeping our projects going and making sure that the students are looked after. I obviously we go to the project meetings together, etc. But she does a lot of the heavy lifting, as well as being a brilliant strategist as well when we talk about where we're going to take our work. So that helps a lot. And then there's obviously a range of people within the department institute that we're able to work together with, and you can sort of share the load. And the centre, I have a centre manager, so that helps a lot.
Wow. Still, it's a lot of responsibilities. You mentioned at the beginning that you trained as a pharmacologist. How did you even get into science?
Well, as a student, science was what interested me. I was never a humanities person. And so, I did science as an undergraduate. And, if I'm being honest here, I ended up doing pharmacology as what I thought as the path of least resistance. It seemed to be the, inverted commas, in my mind, easiest set of subjects to graduate with. So that's sort of why I went down that path. But obviously, having done it, it's an area of research that I'm very passionate about. I believe in concentration response.
Agreed. And I've seen you present, so that comes through clearly. The reason I was asking is I wanted to know if you've always known that you wanted to be a scientist, or was there any other subjects that you were better at? Some guests say, I wanted to be a physicist, but I was not good at physics, and I ended up doing pharmacology instead.
Well, no, my original thought was I wanted to do medicine, but then my marks weren't that great, so didn't have an option of that. But having said that, I wouldn't want to do medicine. Knowing what it is, and a lot of people who are doctors, a lot of my friends are, and so forth, and it's like it's not. I would not have been good at it.
I think you're
not a great people person.
You're not the only one. I think I'm one of those people as well who was pushed towards medicine. I never had the grades. And to be honest, I'm so glad that I did not have the grades. I was more research inclined than medicine. And I think you need to have
so much more interesting.
Yeah, I think so too. And unless, it's very different. I think you have to have a different personality type to do that. Did you think about when you were thinking about medicine? How old were you? I'm trying to figure out, as a teenager or as a child, was it something that you're interested in? Or
I don't think it was anything with a lot of fervor or anything. It was more being a doctor would sound good. It's that sort of level of commitment.
Fair enough.
So basically no real commitment.
Fair enough. That's fine too. I think as long as you went through the path that you went through and ended up where you really want to be, and you love what you're doing, the rest is history.
Yeah.
So
exactly.
When did you first hear about GPCRs?
now you're straining my memory. Probably during undergraduate. I mean, because we would have done beta receptors and whatnot. So almost certainly then. And then my PhD was actually working with a G protein coupled receptor. So certainly by that time I was engaged with GPCRs.
What when you first heard about GPCRs, did you think, oh, this is an interesting system, I really want to get to know more about it, or it was you kind of fell into the field during your PhD?
So the answer is yeah, I fell into it. I was not thinking about GPCRs as GPCRs at all at that point in time, and indeed, what I ended up doing as my PhD was more by inverted commas necessity than particular choice. So I did know from my undergraduate that I wanted to carry on and do research and do a PhD. But my undergraduate marks were not spectacular. I was not good at studying, so I didn't qualify for any of the standard scholarships. And I'll probably make a point of note at this time. I didn't realize it at that time, but I have been clinically diagnosed with attention deficit disorder, which was only about 10 years ago. And I only point that out for those people who are worrying about, they might be maybe atypical, and that there's lots of paths to be successful. So it wasn't an impediment for me. Just meant that I wasn't good at exam type scenarios where I had to sit down and rote learn things. So different paths for different people. Anyway, so because I didn't qualify for the standard scholarships that is the way our system works. So I looked at people who had project funding through scholarships that say funded from their grants and so forth. And so I applied for a number of projects, and one of the places I interviewed, and I'll point out they didn't actually offer the project I applied for, but they did contact me afterwards about a different project. So I guess I at least made a positive impression. And then that would be a collaboration between the group that I interviewed with, which was led by a person called Fred Mendelsohn, who's very well known in that angiotensin field, and also a group at a nearby institution led by Jack Martin, who was a leading bone researcher. And then that turned out to be an ideal project for me because it meant that the technical aspect of my project was led by my main supervisor, which was Fred, and that was using what was at the time a very new technology, which is this in vitro radiography. And the goal was to map the location of receptor binding sites. Whereas the subject matter of my project was actually Jack's interest, which was bone receptors. And in this case, it was the calcitonin receptor. And so, and he was on a different campus, so I didn't see him that often. And that then gave me a lot of freedom within the project. So those within Fred's group working on angiotensin projects, Fred was very hands-on and micromanaged. But, because my area of project wasn't his main thing, I had a lot of freedom to do stuff. And so that was great. And it meant that, I was the local expert, and I could pursue the direction of the research pretty much as I wanted to. It also meant that actually during this time, I actually wrote my first major grant, which was on the work I was doing, which did get funded, but I wasn't an investigator on it because students weren't allowed to be CIs in grants. But that was a good experience as well that I learned during that time, and that came from the fact that it was in a distinct area from the main work of that group. And so, that was formative. That's what led me into studying receptors and studying GPCRs, in this case, the calcitonin family receptor. And then I've been lucky, thereafter to basically pick up independent fellowships shortly after completing my PhD. And then not long after, independent grant funding from the competitive agencies, as well as from industry, and that's an interesting serendipitous story. Might get to that later. And then I moved institutions and eventually went to the Department of Pharmacology in 1998, and that's where I met Arthur Christopoulos. And most people in the GPCR field will be familiar with Arthur. And so we've worked together, obviously, for a very long time, and I've had a very productive collaboration and are very good friends. But I moved from that department to the Florey Institute a couple of years later, mostly for political reasons. And then we moved together to Monash in 2006 as an opportunity to bring our groups together before eventually moving to where we currently are, which is the Faculty of Pharmacy and Pharmaceutical Sciences, which is on a little campus that's actually close to the University of Melbourne. And I mean, Arthur's now Dean, and Denise joined our group just before we moved to the Clayton campus, the pharmacy campus. And obviously Arthur and Denise have been critical collaborators for me. They're amazing scientists, and they've been key people in my career.
And they're next on my email list. I actually emailed Arthur, who accepted to come on the podcast, but I think with the new position he has, it's been really busy.
He's been crazy busy. I mean, he had the joy of taking over as dean just before COVID hit, and so his first 2 years was just putting out fire after fire after fire. Having said that, we're the number one ranked in the world in one of the surveys for pharmacy and pharmacology, so can't be doing too badly.
That's fantastic, and I can't wait to hear back from Denise. I also emailed her. So let us take a few steps back, because you made many great points. First, I want to thank you for sharing the fact that you have been diagnosed clinically with ADHD. I think it's an important topic to talk about, because a lot of us, or a lot of listeners, may benefit from knowing that successful scientists have attention deficit disorder. And that shouldn't stop you from being a great scientist. It shouldn't stop you from being able to have a full, satisfying career.
Well, absolutely. It means you have to work a bit differently. So,
yes.
I'm going to go with that. That makes me more creative. But
well,
I don't think it makes me any more creative than anybody else to do.
I don't know. I think we all have to work with what we're given, with what we have. We can build new habits. We can work around our difficulties and take them as they come. For example, I read a paper and I don't remember most of it. All I remember is the take-home message and where to Google the information. I cannot hold on to information. I can hold on to where to find the information or who to talk to get that information, like you just mentioned. And that's my workaround. And for years, I felt bad about it, because you mentioned Denise and Arthur, but I've had colleagues who remembered all these things. They could tell you the year, the authors, the little details. And in my mind, I was like, maybe it's just me. But it's not.
Yeah, it's it's frustrating. Particularly, I mean, Graham Milligan's great. He has all that information, like snap, 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 and then you'll remember. He loves phrasing like, and you'll remember the paper published in 1993 with these authors, and they did these specific experiments. And we go, yes, Graham, of course I remember.
Yes. And then, how funny that you mentioned Graham, because I was at the GPCR retreat in May, and Michel Bouvier was mentioning exactly the same thing about Graham. And then I recorded our podcast episode with Graham, and he was mentioning specific papers. And in my mind, I did exactly the same thing. Of course, I do remember those papers. But I think it's admirable.
I mean, it's great. I mean, we need people like Graham, who remember, the old literature as well as the new literature, and bringing that up and reminding people is really important. The number of times, for example, that he basically reminded you that somebody had done something already, years ago, and that wasn't that novel. Just is an important lesson, particularly for younger scientists, with so much information that comes out every year. It's new information that a lot of things were actually discovered more than 5 years ago.
Exactly.
And that you need to be aware of that literature, and not just through what's in reviews, because people don't always interpret the information the correct way.
That is correct. That is true. All right. The next great point that you made is really mentioning that you already knew as a PhD student that your working style was not to be micromanaged. You mentioned that you're working on the calcitonin receptors and having this collaboration, having these two mentors, one further out than the other, allowed you to really adapt or to really thrive and be creative in your own project. And I think that's something important that a lot of PhD students or even postdocs sometimes don't realize what is their working style, and that would help them pick the right lab for them.
Yes, I mean that it's important, when you're looking for the lab you want to go into to speak to the people who work in that lab and get a feeling for their style and what works for those individuals within that lab, and also speak to a range of different labs that are in their field of potential interest, because the success is at an individual level is going to be really how much you enjoy the experience, regardless of where you publish the work. If you have a great experience, then you become engaged with research as a process, and that really should be the key driver in terms of becoming successful. If that's not your driver, then your motivation is probably a little bit skewed the wrong way.
What did motivate you to, after your PhD, how come you did a postdoc? Where did that inspiration come from?
Well, it was the natural progression, at least, when I was doing it, that you would go and do, more research, and I was lucky. I had a choice of positions, and so I basically chose one that was aligned to my work that I've been doing, but gave me an ability to learn new things. So that's when I started learning about biochemistry and purification of receptors and so forth. So that was great, and at the same time, I'd had some experience in grant writing and fellowship writing. So I was able to write grants and to get some independent funding quite quickly within a couple of years, so that I effectively then had control of the direction of my research. So I was really fortunate at that level, to have that level of independence that early.
Yeah.
I mean, you don't. That's obviously is probably more unusual than not, and that's certainly not something you need to have in order to be very successful. Building on work that you do in different laboratories in a different setting. But yeah, that was sort of my path, and then there's a bunch of things that happen in parallel that helped. So I'll relate this story. So one of the things that I found during my PhD was that so I was looking at distribution of calcitonin receptors and calcitonin-generated peptide receptors at the time. And these were the only receptors known for that family at that point in time. So as part of mapping where all these CGRP receptors were in the brain, as a pharmacologist, I was also doing competition inhibition studies with different types of peptides. Mostly with different nuclei in the brain have different affinities and so forth. But it turned out that when I was doing this, that there was a subpopulation of receptors that were completely sensitive to a calcitonin peptide. It was actually a fish-like calcitonin peptide, salmon calcitonin. So there was this population of receptors that were totally insensitive and didn't compete at all, and then this other population that were fully competed by this calcitonin peptide. And so that was exciting because it was a novel phenotype, hadn't been recognized before, and it turned out that actually was the first identification of what are now known as amylin receptors. And that then becomes part of a interesting, I guess, serendipitous story. So as a early postdoctoral fellow, one of the things I was doing was collaborating with a physician researcher whose name was Mark Cooper. So he was very much interested in the kidney at the time, and so we were collaborating. I was doing in vitro radiography, mapping receptors in the kidney with him. So it turns out that one of the discoverers of this peptide amylin is a guy called Garth Cooper, who was working at Oxford, but he's a New Zealander. So he was actually the sole scientific founder of what became Amylin Pharmaceuticals. Anyway, so Mark was presenting it at a meeting. Actually, both of us were at that meeting. And Garth Cooper was actually at that meeting as well. He'd returned to New Zealand. This was an Australasian meeting. And in those days, we did presentations with slides. And, so you'd load up your carousel in your prep room, and then they would be taken off. You do your presentation. They all came back. And so Mark ended up getting Garth's carousel. And so he's pulling out the slides and going, what's this amylin peptide thing? And at the same meeting, then Amersham were starting to sell this new radioligand called the iodinated amylin. So we said, well, let's go and have a play with that.
Yeah.
And at the same or very similar time to this, Mark noticed that there was this deal between Amylin Pharmaceuticals and Glaxo at the time for $20 million. And his comment was basically, why don't we see if we can get some of that money? And so, or something to that effect. And long story short, we sent off an email, basically got a response the next day, and we were flown to San Diego the following week.
Wow.
And it was very surreal because it turned out that every single person at the company knew who I was, because their binding assay for amylin were based on that receptor phenotype that I identified in my PhD.
Wow.
And so that was the basis of how they were doing their screening for things. And so we were lucky. We ended up with a bunch of grants from Amylin, from Glaxo, and so that gave us and gave me another block of money that helped build the group quite quickly. So, and that was all serendipitous, but very interesting, very surreal, I have to say, when we went over there, and it was like every person knew who I was. I was like 2 years out from my PhD. So very weird.
It's an amazing compliment, though. And when you were doing those experiments and you collected that data, who would have thought that a couple of years later you'd be famous?
Well, small famous,
but still, I think it's an important contribution. I think you make a great point about that especially for trainees, because, and I'm thinking about myself back in the day. You're so focused on your project, you're so focused on those experiments, but sometimes it's important to keep in mind that one experiment, yes, it might work and it's going to end up in a paper, but others might benefit from that data, and it's exponential, and you don't know.
Exactly. And I mean, that's sort of, in the sort of sets of questions, one of the things you ask about, what are some of the things you would, give as advice to trainees?
Yes.
And the top of my list is like, well, be excited about what you do. We're in a really privileged position that we get to know something for the first time. And, you need to be excited about that. It may be a small, new thing, or it may be a big new thing, or it may be, something that was small and interesting, but actually you learn out later has a big impact. So, that's a key take home. If you're going to be in this business, you need to be, when you see something, get excited.
Yeah.
If you can't do that, then the challenges that are, a reality of our work are going to be very difficult.
Agreed. And since we're talking about advice to young scientists, we can go back to the other questions afterwards. What else do you have on that list when it comes to advice?
So I have this thing and it's probably not the right way of phrasing it, but to be politically aware and engaged, and sort of, there are multiple, potential career paths. But at the same time, there are a whole range of things that you can be proactive about by being aware of what the system requires for different things. And so, there are a whole range of things that you can do to be building items on the CV Like when you're traveling to a conference, there will almost certainly be labs that are worth visiting in the nearby vicinity. And I can guarantee you can basically invite yourself to talk. and people will say yes. And so then you build up this invited presentation and
Yep.
on your CV. So it's little things like that, but also making sure you're engaged and part of the conversation. So that means you got to put yourself out there. Ask your questions. You have every right, but also ability to come up with useful contributions to the conversation. And people need to do that because it's part of being engaged, but also making people aware that you want to be engaged. And yes, it can be a challenging thing. It can be very intimidating. But you can basically train yourself to do this to some extent. I mean, when I was doing my PhD, I was in a department full of mostly medical physician researchers. Most of them went on to become like department heads and leaders in the community. So it was a very intimidating environment. But when we had our seminars, I basically set my task. I would have to come up with a question at every single seminar and ask a question at least once every second seminar. Yep. Part of that just training myself to do this, but also making myself overcome those sort of innate barriers of not wanting to put myself out there that I think most people feel. But we want to know that, and then I think most people experience the fact that if they listen to the questions that are being asked, many times there'll be questions that they had that they were unsure whether they should ask them. Yeah. So be confident in yourselves and get engaged.
I think that's a great advice, and I'm guilty of that. Even now, at the GPCR retreats, there were talks, and I was thinking of questions, and I said to myself, I'm just gonna get the speaker and then talk to them. They have a poster or something like that, and somebody asked the question I thought about, and it's important to push yourself, and me included.
Yeah, and I mean the point you made about at least going up and talking to them later. That's the other thing you can do is get out there, introduce yourself, talk to people. With very few exceptions, people want to talk about work that they do, and they're happy to talk to anybody about it. Gives them a platform. So get out there, do that. Both junior and senior researchers in the field. The one comment I will make is like, don't be offended though if people don't always remember you. For myself in particular, I struggle with being able to recall faces, names, and it's part of that ADD thing. So, but normally if people want to remind me, then I can normally recover the context and remember. But it's like I won't by default likely remember you unless there was a very specific thing like you've done an experiment that I'm really interested in, then that's the sort of information I can retain. Otherwise, I just lose it, and you need to bring me back to it. So that shouldn't stop you from going up and engaging and being part of that. So.
I think you make a great point, and I'm a little bit like you as well. I will remember the context. I will potentially remember the person's face, but sometimes names just escape me. It's like there are times when someone introduces themselves to me, it's like there's a beep going on in my head and it's just immediately deleted. And it's not against that person. And even in my phone, when I save phone numbers, I typically save the name of the person and add something about them, a meeting or a company, so that I can remember where we've met. And certainly you made another great point about our field. I think most people in the GPCR field are always willing to chat, always excited to chat. And yes, sometimes it might happen that people don't remember you and then you have to remind them, or sometimes people are too busy to talk to you. But going out there and talking to people, I think it's just an amazing thing to do, and everyone can do it. It's just a matter of practice.
Yeah, it's intimidating. I that's for sure a reality, particularly for people who are naturally introverted. But there's a reality that most people are happy to talk to you.
Yes. Yes.
I used to, I work with Brian Kabilka a fair bit, and Brian's great and he's actually very funny. But people used to be really intimidated about talking to him because he just doesn't look like he wants to be engaged, but he actually is.
Yeah.
And he's very happy to. It's like, so don't be fooled by if somebody looks like they don't want to. We can't control what our personality reflects.
Yeah.
So, and most people, I mean, you'll get exceptions to the rule, but most people, the vast majority of people, are very happy to have a chat to you.
Yes.
So keep that in mind.
Agreed. And I think even just sending an email or connecting on LinkedIn is already a great first step for those of us who are introverted, who are afraid. But I think to work on that and build that confidence is really going up there at conferences, going to the poster sessions, introducing yourself, talking to people. And even I feel intimidated sometimes. Like, we're talking
I think
now, but
everybody does.
yeah. If we went at a conference, we would talk, but I think I would have to, have enough extrovertness coins in that day to go and talk to you or talk to anyone else. For example, that's also something important for people to understand, that we're all different. We all have our, good times and bad times, and our ability to be introverted or extroverted. I have a number of extrovertness coins during the day, and if I use them up, I'm done. I can't handle more conversations, and it's really tiring at conferences as well.
Yeah. If you're not a naturally extroverted personality, it actually is very, as you say, it's actually very tiring.
Yep.
To actually build the motivation and make those efforts for those of us who need to do that. And I would put myself in that latter category. It's like I don't naturally go and have conversations with people, whereas somebody like Arthur is very good at it. So we're just different personality types, and we and that's actually been great in the sort of relationship that we have as scientists together, because we bring totally different things to the table. And I would happily say that we've been extremely successful as a consequence of that. So that's building networks, building partnerships, and actually that on that sort of point, and not everybody has the same circumstance that makes it easy to do, but if you're able to build strong collaborations, there are a lot of advantages to that. It provides people who can help you if like if your grant doesn't get funded, if you're sort of basically sharing your pooled funding, you're able to maintain momentum of the total research group without being totally reliant on individual grants being successful or unsuccessful. Equally, one of the things that building those sort of collaborations has helped with for other people in our department, particularly for those people who are having families, is that when those people go off on maternity leave, their research doesn't come to a halt, because their collaborative partners then take over the supervision and help progress the projects. So it helps buffer them against the challenges that your circumstances provide. So as well as being actually great in terms of being able to have multiple different types of input and different perspectives on problems. So if there's an ability to build those sorts of relationships, and I very much encourage people to do so, there are a lot of advantages to it. But I understand that not everybody will be in an environment where it's easy to form those sort of alliances.
Yeah. But I think you make a great point about really collaborating and building those partnerships, and doing so at meetings. Now that we're meeting more in person, but even contacting people, talking to people, I think it's important, because it kind of it goes full circle, and you never know who you're going to meet. And even if you talk to someone and you can introduce them to a potential collaborator, that's already a plus, because you never know with your work when you were flown out to San Diego and everyone at the company knew who you were because of that experiment that they used to set up their own assets. You don't know who's going to read your paper. You don't know if you introduce two people how they're going to collaborate. We just, I think 2 or 3 weeks ago, submitted a paper to Science, and it's a collaboration. I'm co-second. We're an army of people on that paper, and that collaboration started out in 2014 at a conference where I was presenting a poster. Someone stopped and said, "Oh, my roommate would love to talk to you." So I ended up talking to the roommates. So I met Ian, who was a PhD student at NIH. We ended up collaborating. He introduced me to his PI. That enabled me to move to NIH later on. And then the work that I've done with them just got submitted. And it was just hey, let me introduce you.
Yeah, I mean, the in-person conferences are major drivers of building collaborations. And, obviously, that's been a component that's been missing. I mean, there have been advantages in these virtual meetings in the sense that it has given access to people who otherwise would not have the resources to be able to get to meetings. But the whole being able to network, talk to people, come up with ideas, start new collaborations, that is obviously something that is an in-person thing that as a major driver for that. So it's good that we're starting to get back to that a bit, albeit that travel is about a third more expensive than it used to be, but
and it's
better
painful.
to be able to travel than not travel.
Yes, but it is painful with all the flight delays that we've experienced. In May I spent the night at Newark Airport because my flight from Buffalo to New York got delayed by 6 hours. So I missed the other one, and it was just this messy thing. But it was absolutely worth it for those 2.5 days of the GPCR retreat.
Yeah, you've got to do a little bit more planning about making sure you've got at least a day spare before anything important, in case you have to deal with those issues. Fingers crossed. My worst delay has only been 6 hours, and I haven't missed any connections.
That's good.
But I don't travel with checked luggage. Doesn't matter how long the wait, unless my wife's traveling with me, in which case she'll have this massive camera and a big suitcase. But otherwise, if it's just me for work, it doesn't matter how long. It's what I can take with me and carry on, and everything else I'll work out post. It just means that you're not worried about connections in the same way that you would otherwise.
No, agreed. All right, let's get back to GPCRs because I have to ask this question. You mentioned working on calcitonin receptors and then on class B1 peptide GPCRs, binding GPCRs. What's your favorite GPCR, if you have one?
Well, it would be a family, and it's the calcitonin amylin receptors. So the calcitonin, so amylin receptors are heterodimers of calcitonin receptor and one of 3 ramps. So there are 3 amylin receptors as a consequence. And so, I mean, they've been, part of critical elements of my career, even though we do a lot of work on other things. So, from my original funding, the original interaction with industry, so it's my first interesting major discovery was around those things. The eventually being able to work out what the molecular basis was for an amylin receptor. I mean, one of the things we had funding for from that original industry interaction was to try and clone amylin receptors. And so our group was doing expression cloning, and where you do, dilution of your things until you isolate an individual. And so we would be tracking receptor binding to a point, and then it would always disappear. And it was nice to learn that wasn't because we were just shit at our job, but there were 2 components, and at some point we were separating the cDNA that was encoding those, and as a consequence, we no longer had the receptor. So, eventually, being able to demonstrate that ramps were the missing link, that was another, sort of a major step. And then be able to, take forward, and so the CTR was actually the first receptor that we did by cryo-EM. It was the first GPCR solved, or at least published by cryo-EM. And then, recently we've been able to solve all the amylin receptor structures as well. So that's been exciting as part of that. So, and they're interesting receptors because, they're now, there's a lot of interest in terms of them as potential obesity targets. And very interestingly, even though we've known for quite a while that, if you activate amylin receptors, you can, lead to weight loss and other energy effects. But the CTR wasn't considered to be relevant to that because if you have selective calcitonin receptor agonists, then they don't do anything to body weight. But the data now looks like if you co-activate amylin receptors and CTRs, you actually get a much more efficacious effect. And so these new dual amylin calcitonin receptor agonists have become, quite a lot of interest around that. And Novo Nordisk, has this compound or drug called cagrilintide. And so they had recent data from that as a monotherapy. They were getting 10% weight loss in patients, and as a combination therapy with semaglutide, a GLP-1 receptor agonist, weight loss around 17 sort of percent. So really, really good efficacy in that sort of space. So I think there's a lot of potential in targeting these receptors for metabolic disease.
And how interesting. And I think I'm trying to think back as to how many people had a family of GPCRs as their favorites, and I think you're among the first ones who thinks in terms of a family. Andrew Tobin
did Andrew only have a specific muscarinic?
Well, no, Andrew had a phosphorylation site as a favorite. He wasn't even a GPCR; it was a phosphorylation site, which I thought was which was, and he was the first person actually who mentioned a phosphorylation site. So we've had people like Michel Bouvier who said, "Well, I don't have a favorite GPCR. I think about GPCRs as tools to better understand GPCR biology in general." We've had Andrew who mentioned a phosphorylation site. We had G proteins. We had people who couldn't decide between the two different G proteins. It was a difficult, depending on who the person is. There is no right or wrong answer for sure.
Yeah, it's a personal question, right?
It is a very personal question.
Yeah.
So exactly. You mentioned metabolic diseases. Do we know about other diseases in which the calcitonin or amylin receptors are implicated in, and what could those be?
I mean, well, calcitonin receptors were primarily of interest in the context of bone disease. And so, for a while there was an interest in peptides and treating osteoporosis and hypercalcemia malignancy. But the reality is it was not a particularly good drug for osteoporosis, didn't do a lot. Didn't matter how many suppositories the Italians wanted to take with it. Didn't make it any better. It was very popular there as a suppository, apparently. Anyway, getting off topic. But I mean, it's still potentially useful for things like hypercalcemia and malignancy, and and some of that relates to the nature of the biology of calcitonin receptors, and so they're more relevant when you've got high bone turnover states. So when organisms are young, or they've got a disease where you're getting high bone turnover, then you can get efficacy with calcitonin agonists that act on those receptors on osteoclasts and basically turn down resorption.
Interesting. Now, you mentioned osteoporosis, and I immediately thought about PTH receptors, and those are also targeted in osteoporosis as well. And I was trying to figure out, okay, what's the relationship between the two, and can we modulate both receptors in order to...
Well, PTH is still a good target.
Yeah.
I mean, there's efficacy for peptide ligands. Though I don't, and it's an interesting target because you have different profiles that appear to be better for some diseases versus others, and there's a lot of complexity in terms of what it is about the nature of the different types of drugs. Is it just that the pharmacokinetics and properties, and and the timing of the stimulation, or is it about the way they signal? So biosignaling and regulation, is that
Yeah.
relevant? Is it targeting subsets of things that might interact with other proteins like ramps? It's a complex but very
Yes.
interesting world.
Yes, and then you have two PTH receptors, and then they have all these differences, and it's this complex yet very beautiful GPCR world,
Well, they're all class B1, so they're excellent.
right? So I asked this from everyone, and I think the answer is always yes, but I was hoping you could elaborate on that. Do you think GPCRs are still good drug targets?
Well, you know the answer.
It's yes.
Yes, of course. Well, if I didn't believe it, so Arthur, Bob Lefkowitz, and myself are co-founders of a company that was just set up recently called Septerna Inc.
Yeah.
It's specifically established on the premise that these remain excellent targets. So
Mhm.
we definitely believe in them and believe that there's a enormous scope there, but there's a lot of complexities in being able to target them. They're not an on-off switch,
Yeah.
and so um, understanding how to target them is often not clear. We can demonstrate that different ligands can do different things, and that's both a big opportunity, but it's also a challenge as to which of those things is what you need to be doing, and is that relevant for all the targeted tissues, or is it different in individual target tissues? And then you've got the secondary problem of having model systems preclinically that are appropriately translatable into people. And very often that isn't a clear-cut translational path. I mean, one of the other receptors we work on, GLP-1 receptors, that's been an area where the translation's been excellent, and so there are excellent peptide drugs,
Yeah.
and now dual peptide drugs and things that target that. Whereas in other spaces, it's a lot less clear, and that's true also actually of the sort of amylin receptor sort of space. Pegrilintide is efficacious, but there have been other peptides that can stimulate both receptors, and they don't appear to be as efficacious at the moment. They've been ligands that are based on a salmon calcitonin template rather than an amylin template that have been less efficacious. But I don't know that that's the reason. I know that they activate amylin receptors differently. We can show that structurally, but I don't know that that's the reason that the translation doesn't occur. And because they all work really well in rodents, which I think is probably due to the fact that you can continue to stimulate aversive pathways in rodents and lose weight, whereas that becomes intolerable in people, or it gets down regulated and doesn't become effective anymore. So I think that, and I'm guessing, I'm not a physiologist. I'm basing this on what my interpretation of other people's work, but I think that's part of it is what the strength of activation is down these different pathways, and what that means for a rodent versus a human. But that's a guess at this point in time. But it points out the fact that there is this translational nightmare that everybody has to deal with. It's not a GPCR specific nightmare, but it's particularly acute with GPCRs, and when you want to do something other than just block it. And so what type of stimulation do you need to target? Just peripherally accessible sites or also central sites, all these things.
Yeah, you made a great point about this black box where you go from GPCR and then GPCR biology and then animal pathophysiology and then animal models, and is that applicable into humans? And I think that's the $1 million question, is can we draw that path out in order to decide, okay, for this is the GPCR that's causing disease X, or at least it's part of disease X. We want to target it this way, and then that's translatable into rodents, animal models, and then into humans. And it's such a difficult path.
Yeah, and obviously, in pharmaceutical companies, there's a lot of emphasis on trying to get some validation in people. So a
Yes.
lot of work around the sort of genomics, bioinformatics, to see if there are naturally occurring mutations from receptors that can provide some validation of it as a target in particular diseases and these sorts of
Yes.
things. But obviously, it remains complex.
It does. Okay, so I have to ask about Septerna. So I'm fully aware of Septerna, and I saw the articles and the news, and it's been in our newsletter. But I would love to, if possible, to get some background story as to how did it come um, alive, and how did you, Arthur, and Bob get together and decide, okay, we're gonna work on this and launch it.
Um, so Arthur and I ended up coming through a different path to Bob. So, and it all converges through this venture capitalist company called Third Rock Ventures.
Yeah.
So Third Rock Ventures is a very successful venture capitalist group. They invest primarily in the sort of biotechnology space, and I think they've established over 60 companies, of which I think only 3 would be considered to be not making any money at all, and everything else is made from little to lots. So they're a very successful group
Yeah.
in terms of identifying space for companies to be set up, and then getting the right people in place. So I mean they were looking at doing this, and they had in the GPCR space, and they had a strategy for doing this. And at the same time, Arthur and I and Denise, for example, we're also talking about doing something similar, and also with very similar strategies using structure-assisted design, using our expertise in molecular pharmacology and so forth. So, and this over extended time period of years. So we started initial conversations with them. Had a whole bunch of meetings. I said over years, decided that it would be better for us to just do one company, and be co-founders in that. And so that's basically what ended up happening. Obviously, a lot of detail in the interim. And then Bob had been involved with Third Rock in the initial setup from their side. So, and that's how we all sort of moved in together. And there are, Arthur, Bob, and I were the sort of named scientific founders, but there are others who are, part of the founder group that don't get the limelight.
That's great. And I've been following Septerna on LinkedIn, so, and Third Rock as well. I'm trying to keep as updated as possible with the GPCR focused companies. One, because it's interesting. Two, I think it's important for people to know that these are potentially options, and even for trainees, you're developing something new. You never know where that where they can take you.
But a lot of the first people in the company, came out of Bob's lab, so or other major GPCR labs. So in terms of the initial science, and then obviously that gets stratified with recruitment of people with industry level experience and in different areas and management and chemistry and so forth. So, but it's built on the back of GPCR science, and it's built on the back of both junior and senior people in the establishment of these things.
Absolutely. All right. I know we're at the top of the hour. I have 2 more questions. Top 3 aha moments that you had as a scientist.
Yeah. So I would guess so the first one being the discovery of a novel receptor phenotype when I was doing my PhD. So I don't know if it's an aha, but it's more shit. That's exciting.
That's an aha.
Without knowing what it means. And then actually working out that ramps for the missing links for making amylin receptors. That was an important and exciting moment. Being able to demonstrate that was actually true. And the last, I don't think I'd call this an aha, but it's more the body of work that allowed us to actually move into the sort of structural biology and cryo-EM space, and now be able to visualize atomic level detail and dynamics of how these receptors work. So it's not so much an aha, but it's
a determining moment.
It's a determining moment, yeah.
Well, it can count as an aha moment. I think it fits into that context as to how your career and your lab and the science that you do.
Less a moment though.
Yeah. Well, it's an aha
more,
decade.
more like half a decade. Yeah.
Last but not least, if and when you do have job openings in your lab and in your team, where can people find you?
So all jobs will go up on the Monash University job site, and we would normally tweet out
Yes.
jobs if they come up. We do actually tend to fill a lot of positions via networks, and they tend to be where we can't do that via networks that we would go to external advertising.
Amazing. Thank you so much, Patrick, for your time.
All right, it's good to chat.
Thank you. Don't go anywhere. I'm going to stop recording and I want to say goodbye to you and official. That's the one thing I forgot to tell you before we started recording. Now, everyone will know that I do that when I record podcasts. All right. Thanks, Patrick. Thank you for joining us and listening to this Dr. GPCR podcast episode. I'd like to thank our team members, Attila Forres, Ines Pinero, and our newest Dr. GPCR proteges, Montserrat Avila Zozoya, and Nipuna Wirusingo. Welcome to the team, Monsi and Nipuna. Please subscribe to the Dr. GPCR newsletter, find us on YouTube, and if you like our podcast, leave us a 5-star review on Apple Podcast. You can also leave us a testimonial at drgpcr.com/testimonials. Another great way to support us is to share your favorite Dr. GPCR program with your network and colleagues. Email us with any questions or suggestions at hello@drgpcr.com, and please become a site member today at drgpcr.com. And until next time, stay safe.